Hello, everyone. Thank you for having me here. So I'm now assistant professor at City University of Hong Kong. After a long time spent in Bay Area, I finally get out of here. So, but I'm happy to be back for a short time. The idea is that we came for many years. We came from 0 interventions to 0 interventions, and the progress is really none. And the question is why? And I believe that's because we don't know what aging is. So if we ask ourselves, you ask different experts in the field, they all will give us different answers. So, and this is actually empirical study, survey, there is no consensus, just, clouds of words. And this is a main limiting factor. The only bottleneck, because some people think that they can cure aging by trying random things or combining different interventions or by applying the papers with omics data to patients. I think it will not work. So in my depressive universe, the gerontology doesn't exist as a field because the main object is not scientifically defined. So there are many theories, hypotheses of aging. Typically people approach aging as a mechanism. So, but unfortunately, the biochemistry is so complex, so many degrees of freedom, that one can build a non-contradictory theory around almost every gene in the genome, around every process, mitochondria, extracellular matrix, cellular senescence, whatever. It will not be will not sound crazy. But to test and refute this hypothesis will take a lot of time. Only this oxidative hypothesis of aging took decades to be found wrong. So that's why I believe this is a very difficult path to go. And it's clear that we can produce 300 more hypotheses of aging very easily. So what my approach is, that aging is, first of all, ecological phenomenon. And so, to understand something in biology, we need to think about evolution. And if you look in the animals, we see that animals age in a very strange way. Some animals die soon after reproduction, like these Pacific salmons. Some, like bats or birds, are living so long for their sizes, it's not clear why. So naked mole rats barely age at all. So in different cases of eusocial insects, age at rates of 50x. So, and we don't know why it's the same genome. What happens there? So and the classic model cannot explain any of these effects consistently. And there are only 2 to 4 evolutionary hypotheses of aging, and they all are somehow disconnected with reality if you look deeper. So, and I'm trying to build an evolutionary paradigm that can fit all this data, and I'm quite successful on that. So what the first question we ask from our paradigm: what is aging? Is it damage accumulation, or it's a program that is somehow in our genes that kills us? So these are 2 very different paths, and they largely define the approach we use to cure aging. Because if it is damage, then we need to repair this damage, help these dots to fly back on their spots, or at least slow down their dissipation. If it's a program, we need to find these genes and this gun and remove the bullets. Right, so in one case we need to fix something. In another case we need to break something down. These are 2 different things. If we pass this fork in the wrong direction, we probably lose all the time and resources. So most of the researchers here, I think, believe aging is about entropy. However, this point of view is not proved experimentally. There is no experiment that would say aging comes because of damage. So all the experiments have been done so far can be rationalized in the programmatic paradigm. So why do we stick to damage? Because we don't believe it can be a program. Why it cannot be a program? There are 2 historically raised arguments that are considered very, very strong. So first of all, if aging is a program, what can it be good for? So idea of evolution is to produce babies. If I die of aging, then I cannot produce babies. Clearly cannot be good. So second argument is, why don't we see immortal mutants? If aging is in our genes, then we can mutanize these genes, get an immortal Voldemort, and we never see it, neither in nature, neither in humans, neither in the experimental animals that we can feed with mutagens very, very well. So cannot be. So, however, I have, to make a long story short, I have made a model that can deal with these arguments. So let me introduce you to this model. So basically, I found that aging can be beneficial in epidemiological models. Imagine a population of animals, let's say this fish, and a parasite. The immune system of the fish cannot clear the parasite out. And let's say the probability of being infected is constant over years. So, and with this, the older the fish is, more is probability it is infected. So at certain age, like in this case, it's age of 8 years, every fish has a parasite. So this particular parasite doesn't sterilize animals, so they can reproduce, but many of them do. And let's say after a certain age, every animal is sterilized by parasite. It cannot reproduce anymore. So for its evolution, it is dead already, because it will die at some point for some other reason. But it cannot produce babies, so it is, its value is 0. But it can infect other fish that swim nearby. And these other fish can be its relatives. So it not only cannot propagate its genes, but it even can damage its own kin. So because of that, this fish has a negative value for evolution, and evolution may want to kill these fishes. So because it can be tricky to discriminate between healthy fish and infected fish, evolution may want to kill them just based on the statistical expectation of being infected, like as a prophylactic measure. And this why I believe, how I believe aging evolved. So we can easily model that. It was a paper in PNS some years ago. So these are yellow one are short-lived animals, red ones are long-lived animals. So if we run the story, you see that black dots are infected ones. You see that in short-lived population, the epidemics propagates not that much, but in this long-lived population, epidemics propagate strongly because the animals spend more time in the infected state. However, if, of course, if we put a long-lived mutant in the short-lived population without pathogen, it, of course, takes over because they live longer, propagates, leave more progeny, and long life is generally adaptive. Now, what happens if we put this long-lived mutant into a population of short-lived animals in presence of pathogen? So see, first it expands, then it encounters this parasite. Parasite selectively sterilizes it because animals spend more time in, we can run it again if you want. So the animal spends more time in infected mode, in an infected state. And this actually is a basic model of selection towards shorter lifespan. Now, so we figured out why aging can be beneficial for evolution. Now, what the heck with the immortal mutants? Why we still don't see it? Okay, why some genes that are involved in adaptive aging, if we mutagenize them, they don't give us Voldemort. We clearly see what we can interpret as a gain-of-function mutant, this progeria, but we never see immortal ones. So this argument is not entirely true because we see naked mole rats, and naked mole rats share common ancestors with these other rodents that age. So at some point along this branch, like aging mother should have produced non-aging or almost non-aging pup. So it is possible to make an immortal mutant, but why doesn't it happen so often? So why aging is so well fixed in our genome? So let's look in the cases when aging is not very well fixed. Let's say these eusocial insects, they have short-lived castes and long-lived castes. Queens can live 50 times longer than workers. So apparently parasites can switch between these programs. Parasites want us to live long. So because they can propagate, right? They don't want us to die. So, and in this case, the parasite, the tapeworm, can obviously switch the program that short-lived worker start living like a queen, so 50 times longer. So parasites want to inhibit aging, paradoxically. And and there is evidence. This is not only the only evidence, actually, because also in humans, some parasitic nematodes can reduce inflammation and reduce mortality. So idea is that aging is fixed with some safety harnesses that prevent parasites from manipulating it. And these harnesses prevent not only parasites, but also effects of mutations and treatment. That's why it's so difficult to inhibit aging with interventions. So we have solved both of these questions, and now I believe there is no reason to believe in damage anymore. So we need to switch to program models, and I need to say that my model can explain all these paradoxes, like correlation between aging and flight, why naked mole rats, what are the ecological reasons for longevity in naked mole rats. A classic model cannot explain that, as well as what happens in why eusocial insects or why simiparous animals do that. So there is no time to go into detail about all the aspects of the model. It's quite developed. So, but the take-home message is that out of all aspects of aging, immunity is probably the central one. It is sort of instructive. So, for example, if this cellular senescence is also not clear what's good for, right? So the cells undergo this senescence and become toxic. And why evolution developed that? So, and we believe these senescent cells are also part of the immune system, and this we can do. So in this, look at this video. So we infect senescent and control cells with GFP-labeled vesicular stomatitis virus. And you see the difference, right? So senescent cells are antiviral. And this is true for many other viruses. So these are, you may not see it well, but these are like viral production in control cells, 5 different viruses, 4 different families. In senescent cells is much less. And this is not because the senescent cells are so weak and sick. So because if we add inhibitor of JAK-STAT pathway, we rescue the virus reproduction. So also we have data that SASP, that secretion that senescent cells produce, can protect healthy cells. And so this brings us to this immune militia hypothesis of cellular senescence, that actually healthy cell, if infected, it can be conscribed to the immune militia, so it like to protect the body against infection. With SASP, it protects healthy cells, communicates to professional soldiers, which are like immune cells, normal immune cells, and after infection is resolved, these cells need to be demobilized because they still can contain some virus that can reactivate. And this is I believe that this is one of the main functions of the senescent cells. If they are not cleared, they start to loot, of course. And yeah, so this is sort of a fundable spin-off of my big model. So, but there are also conceptual crazy projects, because I tell this model to different scientists. They say, what have you done to falsify your theory? So actually, to falsify, so it is difficult to confirm or refute the big evolutionary model. But we produce some crazy predictions that are potentially testable. So first of all, why do mole rats live so long? What are the ecological reasons? And the prediction is that their eusocial structure, that they have queens and workers, they can allow a very unusual strategy of controlling disease. If the very first animal that is infected dies by sort of adaptive suicide, anaphylactic shock, then the colony can be kept clear from pathogens. And if so this should work much more efficient than aging in controlling pathogens, and then aging can become redundant. And so prediction is that naked mole rats should be hypersensitive to any infections, and there are like case reports so that people try to put gene of interest into naked mole rats using herpes simplex virus as a vector, and all these naked mole rats died. So very sad case report, sort of don't do like this. But so the idea would be to test this experimentally, find mechanisms, and test the prediction number 1. Crazy prediction number 2 is about similparity. You probably don't know this word. It's not aging, it's a type of death that animals undergo after reproduction. Like the classic example is Pacific salmon. Salmon goes up the river, lay eggs, die. So in mammals, it's almost absent. However, in carnivorous marsupials, it evolved like 5 to 6 times independently. Why? What are the these mysterious ecological reasons that predispose carnivorous marsupials to similparity? And the idea is they found that the model predicts that there should be some unusual disease going there. And we believe this can be transmissible cancer. You probably have heard about transmissible cancer in Tasmanian devil, and this is a disease that have epidemiological properties that can indeed stimulate evolution of similparity according to our model. So the prediction here would be to find more transmissible cancers in other species of carnivorous marsupials. Ecologists do not believe that, but so meaning that if you find it, everybody will say wow, and maybe raise their eyes from their own plates. So with this, I shut up. So this is my lab at CTU. These are people who worked with me at UCSF, and I'm happy to take questions.
All right, folks, let's have a few questions here.
I'm dredging my evolutionary studies memories, but there are pathogen control or infection-related theories to do with the evolution of sex. And sex has an enormous genetic cost in this, essentially you dilute your DNA by 50%, right? So my question or comment is, it strikes me actually, if one already accepts that sex has evolved as a pathogen control strategy or pathogen resistance strategy, this might actually feel like a rather modest proposal in that context.
Yes, so it is not well accepted. There is still battle about sex. We also have our own ideas, of course. So, and I definitely believe that sex is about pathogen control. So the you mean this that we have two genders, like boys and girls do not have male genitalia. So this, I think, is because of the pathogen control.
Okay, next.
Yes, thank you very much. So I'm curious, if your theory is correct, could a fairly benign parasite, but technically a parasite, be engineered that would have the kind of protective effect against aging that you described, so that humans could ingest that parasite to overcome any hypothetical aging program or the safeguards that keep that program in place? Could that be the solution to aging potentially, in your view?
I'm afraid that no, we are not. And so it would be possible if we would have like, if aging wouldn't be that fixed in us. So if we would have two programs like the queens and the workers, then yes. But we still can learn a lot from interactions between parasites and aging. And one of the part of my research program is to learn how viruses are interacting with cellular senescence and try to understand the biology of cellular senescence better and instruct maybe senolytics based on that. Yeah, but unfortunately, the parasites cannot help us to grow wings, right? They cannot produce new function. They just can exploit what is in us already. So the potential of our lifespan plasticity, and it is unfortunately limited.
Okay. Do we have a last question? No? Oh, there we go. Here in the back.
So over the last 100 years, we've removed large selection pressures on our age. Do you think that means our baseline age will change now that that selection pressure is gone without any other interventions? Is that a prediction?
Yeah, in a couple million years, we will probably become mole rats. But I think this started much earlier than last 100 years because just our cognition of the intelligence helps us to avoid pathogens very well. So for 7 million years, we doubled our lifespan, being a long-lived species with small population numbers. So this is very impressive. So our separation from chimpanzee was 7 million years ago. So chimpanzee lived 60 years, we lived 120. So I think we are quite good already in controlling pathogens just by behavioral methods, like, you know.
All right, so one final question I have for the audience, actually, before we wrap up here, is raise your hand if you believe we have a good theory of aging, if you personally believe in a specific theory of aging. Anyone, regardless if it's this one or another. Raise your hand if you believe there is such a thing. Okay, well, clearly there seems to be some need for theories of aging, or maybe there could be a gap there. Anyway, thank you so much, Peter. Give it up for Peter Lidsky.
Thank you for having me again.